Hello everyone, myself Dr. Mahesh JoshIi. Today I will explain structure and functions of Golgi complex. First of all, introduction. Golgi complex is a stack of membranous flattened sacs and vesicles concerned with cell secretion. Golgi complex was first discovered by Camillo Golgi in 1898. Most of the discoverers or most of the scientists said that Golgi complex were arises from the endoplasmic reticulum. Golgi complex is also known as Golgi apparatus or Golgi bodies. It is located in cytoplasm. It is membrane-bound cell organelle. In animal cell contain only one Golgi body. But developing oocytes of chordates, nerve cells and liver cells contains many Golgi bodies, and also plant cells usually contains hundreds of Golgi bodies. Golgi complex is absent in prokaryotic cell. Certain fungi, sperm cells of bryophytes and pteridophytes and RBC. Structure of Golgi complex. Golgi complex is a stack of membranous flattened sacs and vesicles interconnected by tubules. It has variable in size, larger and well developed in gland and nerve cells, as well as poorly developed in muscle cells. Larger and well developed in gland cells and nerve cells, it means that it is well developed in pituitary gland as well as in thyroid glands. It has disc-shaped structure formed by a stack of cisternae and peripheral vesicles. This is the figure. This figure shows that this is cis phase and this is trans phase. Cisternae. These are the cisternaes and these are the vesicles. Okay. So this is cis phase. Cis phase is lies towards the nucleus and endoplasmic reticulum, and this is trans phase. It is this trans phase is facing towards the cytoplasm. Okay. And this trans phase is far away from the nucleus and endoplasmic reticulum. These are the secretory vesicles, and these are the newly forming vesicles. Golgi complex, it consists of two parts. First one is cisternae. The cisternae are elongated flattened sacs of Golgi complex, filled fluids with and peeled one upon the other to form stack. They are arranged in parallel bundle of one above other. In most cells, number of cisternae varies from 3 to 7 in a stack. Other cell types may have as many as 10 to 20 cisternae. The adjacent cisternae are cemented together by a cementing substances called as intercisternal material. The Golgi complex has two sides, namely cis phase and trans phase. The cis phase is lie towards the nucleus or ER. This phase is the convex surface. The trans phase lies towards the plasma membrane or far away from the nucleus and ER. This phase is the concave surface. In this surface, large secretory vesicles are bundled off. Second point is vesicles. These are the small droplet-like structure of 40 angstrom unit in diameter. With the periphery of the cisternae, vesicles are closely associated. They develop either by budding or by constriction of the ends of the cisternae. The vesicles are of two types, namely secretory vesicles and transporting vesicles. Secretory vesicles, the vesicles that are transfers from one cell to another cell are known as secretory vesicles. Second one is transporting vesicles. The proteins that are released from ER and that enters into a Golgi complex in the form of vesicles are known as transporting vesicles. In the Golgi body, these vesicles are modification. In the Golgi body, these vesicles are modification, receiving, sorting and shipping of vesicles into the target site are carried out by Golgi body. Functions of Golgi complex, they are known as post office of the cell. They function as the receiving, sorting, distribution, shipping departments of the cell. Cell secretion produce lysosomes, pectin and cell wall of the plant cell are produced by Golgi body. Biogenesis of plasma membrane, that it means that they helps in the formation of plasma membrane. Secretion of mucus, saliva, sweat glands, oil glands, oil, tear, enzymes, hormones, antibodies, milk and lactoprotein by mammary glands, melanin pigments, collagen and neurotransmitters. Transport of macromolecules within the cell and across the cell in the form of vesicles. Acrosome and cell wall formation. Glycosylation of lipids and proteins. Processing of hormones. Thank you.